Hi, thank you. I'm John Grime from the University of Chicago, and I'm here today speaking on behalf of Greg Voth's research group. So, what I'm going to be talking about today can be grouped under two major aspects. We use the Blue Waters facility for many things, but with the limited time and to keep things cohesive, I thought I'd just focus on two particular elements. So, we first and foremost use the Blue Waters system to develop novel high-performance computing software. We have specific reasons for needing our own software in many cases, and the Blue Waters facility is a great example of how you can integrate the science teams with the technical staff in these facilities to really try and produce something new and useful. So, we're interested a lot in coarse-grained simulations, and with a coarse-grained simulation, you'll take molecular components of a system, you'll reduce the level of detail to simplify those molecular components, and hopefully by doing that, you make it much more computationally efficient, and this lets you study much larger systems over much larger length scales. And to be able to run these sorts of simulations, certainly the sorts of simulations that we're interested in, there are certain specific needs that we have, and we refer to this as ultra-coarse-grained MD. So in an ordinary simulation, you'll have molecular components that are maybe static in their properties. They have interactions that are defined in advance. They have structures that are defined in advance. For our purposes, we want a great deal of mutability inside the system. We want to be able to switch the molecular properties at runtime to enable us to study certain processes. And to make this work well, we clearly need the dynamic system components, but we also need low memory requirements. If we want to push the simulations up to very large scales, we need to be able to make sure we're being careful with what we do with the memory. We need to be able to do dynamic load balancing very well, because for many biological systems, if we coarse grain in such a way that we remove the water, we're left with large empty regions inside the system, and actually balancing the calculations that need to be done across many compute nodes can become a little tricky at that point. Flexibility is good, because what we think we need in advance is never quite what we need in the end. Anybody who's actually developed these simulations in the software will be familiar with the idea that you can't just fix things at the outset. Things are going to change. And so, fundamentally, with the biomaterial simulations we're interested in, sort of cell-scale processes, we can span many orders of magnitude. The interactions themselves may have angstrom or nanometer resolution, but the size of the systems that we're studying may go up to microns, for example. So you have this very large length scales that we're spanning, so we need to be careful about what we're doing with memory. For one example here, anyone who's familiar with molecular dynamics is familiar with the idea of a Verlet list, where you make a list of things that are possibly close enough to interact, and they will remain close enough for a few time steps. And the way that you generate the Verlet list is typically using a link cell algorithm. But if you have very large empty regions, the conventional link cell algorithms are just wasting memory. You're sort of covering empty space with data where it isn't needed. And so we take a different approach for these sorts of systems by using sparse data, where we generate memory usage only where it's actually required. And by doing this, we become pretty efficient in terms of the memory that we're using. To actually do the load balancing, we took a trick from the astrophysics people that we knew, and we used space-filling curves, which are very good in terms of condensing calculations in local spaces that maybe are separated by large voids, such that you can balance the work out across many compute nodes and do so efficiently. But the primary science drivers, the reason that we generated these sorts of software tools, are typically the biomedically relevant systems, because we do enjoy working with experimental colleagues in various places to attempt to add some sort of value to experimental data with simulations, either by helping to interpret experimental results, or possibly trying to access aspects of the system that you can't easily get to with conventional experiments. So I'm going to quickly go over a couple of use cases here, one of which is the viral life cycle of influenza, or a specific part of that, and also with HIV-1, again, specific parts of the viral life cycle to which we're applying these simulations. First and foremost, influenza budding. When you have a cell that's infected with influenza, to be able to spread that infection to other cells, you have this sort of aggregation of viral components at the membrane of the infected cell, which then sort of balloons outwards to form these spherical viral particles that eventually will come free from the cell and go off to infect other cells. And one of the important parts of this influenza system is the formation of this stalk. It's like a membrane neck that connects this viral particle to the cell before the viral particle is released. And influenza is interesting because you just have this one protein called M2, which is sufficient to actually remove that neck. There's some sort of cleavage occurring in the membrane to release these viral particles. So it's a very simple model system of how you might look at these viral systems. So this work was done by Jesper Madsen at the University of Chicago, and we're looking at ways of generating stable budding conformations. There's certain theoretical work around that says that these intermediate conformations between a flat membrane and a completely free viral particle are unstable. So we've had to try and figure out ways of generating these conformations in the molecular simulations such that we can stabilize them and examine these intermediate stages to see what's important for their stability. And we've generated models that allow us to do this by using a sort of polymer brush approach and forcing it through the membrane to stabilize it, and then fixing the membrane size. And this gives us a nice, stable, intermediate structure, and we can change the membrane area, so we can move the budding structure up and down to sort of reverse and go forward in time to see how the actual temporal correlations in the membrane properties and the actual protein locations help us with this. So we're looking at things like the curvature of the neck in the budding region and how the model M2 proteins are attracted to these regions of local membrane curvature and how they then go on to destabilize the local membrane. In a similar vein, albeit in a different system, we're also interested in the budding of HIV-1, which is similar to the influenza system, but it has important structural differences in terms of the molecules that are involved. So in terms of the HIV-1 viral life cycle, this can be considered like the initial stage when the infected cell is trying to release these viral particles. So we have RNA that gets aggregated at the membrane in the presence of these so-called GAG polypeptides, which are associated with the membrane. And this sort of agglomeration of molecules leads to the deformation of the membrane and this sort of hemisphere that starts to emerge. And there are very complicated arrangements of molecules at work here. Here's some experimental data, and you can see that there's a distinct hexagon motif in terms of the orientation of these molecules. They come together, they form local hexagons, and these local hexagons are interconnected. And we believe that the shape of these viral particles as they bud is actually driven by the distribution of defects in the hexagonal lattice. And to look at this in a computational sense, we generated coarse-grained models of the viral components in a very simplistic way. So we have a polymeric RNA, and we have the sort of coarse-grained version of the GAG polypeptide associated with the membrane. And the coarse-grained model is capable of self-assembling under the influence of the RNA to reproduce this complicated hexagonal packing and drive the deformation of the membrane. And we've compared the results of what we see in terms of the local structure to the experimental data to a high resolution, or as high as we can get with the experimental data. And we're finding that for even a pretty simple model, we can reproduce most of the important effects with this, and we can get a good agreement with the actual structural data. And in collaboration with Jennifer Lippincott-Schwartz's group at the National Institutes of Health, we've been using this model to study the influence of so-called microRNAs. So we know that when the cell overexpresses small pieces of RNA, it will interfere with this process of the molecules aggregating and causing budding. So when we just have one long strand that's akin to the actual viral RNA that's packaged inside this budding system, we can get these nice big islands of contiguous lattice to form. But in the presence of the microRNA, it jams the system up completely. We no longer get this nice, large regions of the hexagonal lattice to form. Another part of the viral life cycle of HIV that we're interested in is the formation of this so-called capsid structure, which is this cone-shaped green thing here. And this, again, is an example of self-assembly. We have many copies of a dimeric protein in solution, and these pack together in a very specific way to assemble these structures that are composed of both hexagons and pentagons. And it's important to note at this point that the hexagonal structure of this cone-shaped structure that you see inside the viral particles is very different from the immature lattice. There's a great deal of reorganization that goes on in the maturation process to take us from the immature lattice to the mature lattice. So we're interested in knowing more about how this structure self-assembles inside the viral particles. And to do this, we took note of the fact that the experimental data suggests that these dimeric structures in solution are actually very dynamic. The two different domains of the protein, which is shown here in green and gray, they're sort of tumbling around in an uncorrelated fashion inside the solution, and we wanted to somehow incorporate that inside the model. And to do this, we made a coarse-grained model that includes both a crowding agent, because the interior parts of these viral particles is full of all sorts of molecules. It's a highly crowded system. The idea that you just have these proteins floating around a solution on their own is not really a good reflection of the true nature of what's happening here. And we modeled these proteins as dimers in solution. Now, importantly, what we've tried to do with this system is to incorporate the idea that these domains are tumbling around in an uncorrelated way in solution. And to do this, we had a simple two-component system. So if we consider we have dimer type A can aggregate with A and sort of self-assemble into these larger structures. But we have a B-type dimer, which is representative of the fact that in solution, many of these structures are not actually assembly competent. And by mixing A and B together and randomly switching the background proportion of B to model this sort of transient tumbling of the domains, what we can do is we can control the self-assembly of the capsid protein. Now, inside these viral particles, there's a lot of capsid protein. There's way more than you need to actually generate those cones. So this is one way of trying to figure out what's going on inside the viral particle that will give you the controlled growth of these systems. So in the absence of our sort of background switching of the conformational states, we just get uncontrolled assembly. Everything starts nucleating and growing all over the system. You can't control it into a single capsid formation, and it just turns into a mess. But in the presence of this sort of UCG model, where we're changing the nature of the components in the system dynamically at runtime, we find that, it's easier to get single nucleation and growth events that we can then use to study how these small regions of capsid lattice are sort of nucleated and growing, and how we sort of end up with these curved shapes, because the curvature all over this lattice is quite different, and yet it's made of the same components. So something is breaking symmetry, and this allows us to try and investigate some of the properties of the system to try and figure out why it would give us certain conformations. And what we found through these systems was a few interesting things. First of all, in solution, you get a transient population of these little triangles that are formed from 3 dimers that come together transiently, and then they'll dissociate. So you have a background population of these little triangles. And the little triangles form the basis of the nucleation of these larger structures by the addition of further dimers around the edges, until we find that we have a hexagonal shape in the middle that's stabilized by the sort of skirt of the little triangles. And once we have these structures appearing, they're stable. Up until this point, it's metastable. When we have that sort of a structure, it stabilizes, and you get the concerted lattice growth to occur at that point. So that's a whistle-stop tour of the sorts of things that we're using the Blue Water system for. It's both software development and using that software to investigate biologically relevant systems. And obviously, we need to thank our experimental colleagues here. We have friends at the University of Virginia, Caltech, and at the NIH. And we'd like to make a special thanks to Robert Bruner, who's been our sort of long-standing point of contact at Blue Waters, who's really helped a lot in terms of the technical aspects of this, using the low-level communications facilities in Blue Waters, like the RDMA functionality and so on and so forth, to make our software efficient and to do performance testing and debugging and various things. Also, a big thanks to the Cheetah NIH-funded center that's been helping us to put our work together with the experimental guys and helping to partially fund some of the science that we're doing.